on behalf of the International Science Council and also on behalf of the African Academy of Sciences and the International Development Research Centre Canada. This is an open session of a strategic retreat that we're having over the 2 days to explore the impact of emerging technologies on science systems, and especially those science systems at home in the global south. Yesterday, we discussed some of these emerging technologies. It's not an exhaustive list, but we discussed artificial intelligence, data management and storage, virtual and extended realities as a technology, and also looked at various types of emerging connectivity technologies. Today, we have the privilege of hearing from 4 of the fellows of the African Research Initiative for Scientific Excellence, ARISE program. And we will hear about how they are using some emerging technologies in their research work on the continent of Africa. But before we embark on this, we thought it would be a great opportunity to check in with you as the wider audience and get a pulse of your ideas on emerging technologies. So going to share the screen, including a QR code here to a short Mentimeter survey where we'd love for you to join and connect and give us your feedback. So if you can scan this on your phone, or we'll also have the link in the chat. If you're online, please join us. The first question we'd like your input on, and a question that came out of our discussion yesterday, was what do you understand by an emerging technology? We'll give you a couple of minutes just to put a few words in as an answer, and hopefully a word cloud will appear on the screen. Thank you for that one person. A couple of words are popping up. Untested, interesting, needed, novel, developmental, innovative, AI, certainly coming up as an emerging technology. Disruptive, that's a good one. Brilliant, we've got about 25 people connected to the Mentimeter, and slowly building the word cloud. We'll give you guys another 20 or 30 seconds. Problem solving, exciting. Certainly been some exciting discussions over the last day. Socially salient. It's really good to have this input, because we got to the end of yesterday and thought there's not a shared understanding in our room of what an emerging technology is. Please see the link in the chat. I think we might have to repost it. We're just doing an online exercise, getting your perspectives on what is an emerging technology. Great. Well, let's move on. There's one more question, just to warm your brains up for this morning. We like to get a feel for how these 4 technologies, the ones we discussed yesterday, how they compare in terms of adoption and their potential impact on science systems. So you're asked to rate them highly if they are highly adopted, or if you think they will have a huge potential impact on science systems, and low on the scale if you think their adoption is currently very low, or if you think that their potential impact on science systems is not really that great. So as you move those sliders around, we should see these 4 points appearing on this plot. Interesting. I think what we see, impact on science systems with virtual reality and in the trailing. And then we have a kind of a spread of current adoption, but probably because this was one of the issues that we struggled with yesterday. You know, where is adoption high? Where is adoption low? If adoption was high, this wouldn't be an emerging technology. So there's a lot of kind of an idea of the spread of these points. It's going to be very useful. We're just seeing an average now, but it will be really useful for the, you know, we've got about 87 people online, probably 30 that are filling in this, this little poll. And so I think just having a kind of a snapshot of this will be really useful for the output and contextualizing what we mean. Great. So thank you very much for being a participative audience today and for stepping up to the challenge of helping us with our definitions. I'd like to now stop the screen sharing, and I will hand over to my colleague, Doreen, who will be introducing the speakers for the session. Thank you.
Thanks, Vanessa. So indeed, the session of right now is called Technologies in Action. This is a session part of this bigger 2-day meetings that we're looking into emerging technologies and their impact on science systems. We have 4 speakers today who will let us know how they're using technologies in their research. The session is joined by online participation and in-presence audience. So we'll be taking 1 to 2 questions after each presentation to keep us interactive throughout this session. So please, both in-presence audience and online, you can raise your hand, and you can also add your questions in the chat for online participants. My colleagues will relay your questions to me. So our first speaker today will be Dr. Amna Hariga, a researcher at the Institute of Pasteur in Tunis, in Tunisia. We'll be sharing the slides now for Dr. Amna, and the floor is yours.
Thank you, Doreen. Good morning, everyone. So waiting for the slides to be up. So today I'm here to, my name is Amna Hariga. I'm a researcher at Institut Pasteur of Tunis, and I'll be giving a talk about the research that we have been conducting as part of the ARISE program, supported by the African Academies of Science and the European Union and the African Union. And it will be about artificial intelligence for drug discovery. So it's the main field that we will be discussing today will be composed as computer-aided drug discovery approaches the structural bioinformatics, a broad field of application, where the idea is to reduce cost and time, and also to bring insights that are otherwise unable to be performed through experiments, through molecular modeling approaches. But nowadays, with artificial intelligence, we are able to even lower the cost and time of the process, but also enhance the outcomes. That's what we will be talking about today. So I'll be sharing some insights from a report that I have been part of and that have been conducted by Wellcome Trust about the landscape of AI for drug discovery, launched in 2022. And we have been seeing that there were not much companies that are AI first biotech in the world, and that much of the investment have been invested on only 20 companies on this. And here we can see how things would be split in terms of investment by type of applications. And today we'll be talking about the small molecules, and you can see that there are up to $4 billion invested in that specific field. But when we look closer, we would see that there is a big and huge gap between adoption and use of AI in drug discovery between high-income countries and low-income countries, with very up to 52% of no adoption in some countries, in low-income countries in the world. This is also a gap that we can see between applications in oncology and other type of diseases compared to infectious diseases, and mainly infectious diseases of poverty. And you can see that only 5% of the projects or research done worldwide on small molecules are interested in infectious diseases altogether. So this is where our project and work comes, and we have already assessed and saw that most of the impact can be done in terms of drug repurposing, especially against neglected diseases at the early stages of drug discovery. So here I'm presenting an overview of what an AI-based drug discovery pipeline would look like. This is a very classical way of seeing things. Everything starts with data collection and cleaning. Then we will be encoding data to make these information, chemical information or biological information, something that the algorithm would understand. Then we will train and validate deep learning model or machine learning model in order to provide either prediction of biological activity, or also generate de novo design of molecules for drug discovery. So that's the scope of our work. We have been starting doing this research since 2018 in our lab. And we have gained through a lot of research that have been focused on infectious diseases, mainly COVID-19 and leishmaniasis. We have gained a lot of perspective on how the data integrity or heterogeneity can help or not in advancing the and leverage the outcomes of using AI for drug discovery. So moving forwards, we have leveraged in the frame of the ARISE grant this financial support in order to implement the BIND project called Bioinformatics and Artificial Intelligence for Drug Discovery Against Infectious Diseases. And it has two major subcomponents. First, applied drug AI for drug discovery, where we will be presenting successful projects we conducted against leishmaniasis in particular, but also I'll go through innovative approaches that we are developing in my team in terms of novel AI approaches. So I'll tackle things in three parts of this presentation. First, we will talk about data. As you have already seen, the data is the starting point of any AI project. Then we will talk about applications, which is the third part, and then we will come back to the second part, which is the development part of AI. So first we started to leverage whatever existing in the literature against infectious diseases, mainly leishmaniasis, but also then we had COVID-19 that came in the pipe in 2020. And we have used multiple type of approaches, including computer vision, in order to extract information from the literature in order to implement a database that have been then enriched with information from public resources and databases in order to have a very specific and curated data set against leishmaniasis and coronaviruses, both containing more than 3,000 molecules, which is not much. But this type of information have been used to enrich existing large data sets that are there that are open source. Such a small amount of information have helped deep learning model, but also machine learning model, have a higher impact and performances overall on both diseases. So we have then implemented CDLsDB, which is a platform that is open and can be used by the community in order to browse for molecular entities with anti-leishmania or anti-coronaviruses biological activities along their chemical information encoded as SMILES. They also, all researchers, even if they don't have any coding skills, will be able to predict any novel entity that they are interested in or working on in their labs, to predict their activities with different machine learning and deep learning models. This work have been published, and the CDLsDB is there and running if you would like to test it. But how does this translate to real drug discoveries? In fact, we have been doing this alongside with other approaches that are into the pipe, not yet published, where we have been able to overall predict 71 active molecules, out of which 27 have been validated against the literature to be active against leishmaniasis. The 40 remaining have been purchased, and out of 24 tested molecules in the lab, we have 9 novel molecules tested against leishmaniasis with approved activity against the parasite. Out of them, 3 are very promising drug candidates for patenting projects. So here I'm presenting some of the research that already have been published in 2024, and we have some additional molecules that are under preclinical studies in order to really go from there into directly the third phase of the clinical studies, since this is drug repurposing, and there is a lot of discussions with the ministries and other stakeholders in order to have a good translational process of our research. Here I'm just presenting like a sort of a new sub-project that came up. And the idea was to leverage computer vision in order to automate and be quick on the drug discovery part through using deep learning models for object detection to count and analyze microscopy images throughout the drug discovery project. So that's a very different type of application that we're using of AI, and the idea is to make it also useful as a mobile application for low resources settings and laboratories that do not have high capacities in microscopy and image analysis. So the bind is not just leishmaniasis. We also have projects on flaviviruses that are going on. Toxicity predictions of the molecules in order to have optimal predictive profiles of our molecules at the early stages, and we have collaboration on malaria and cancer going on soon. So the last part of my talk is about, this is one of the projects where we are more into the development of new frameworks in AI. I just chose one of them, not to be very long. And here what we did is that we integrated a quantum circuit into an established tool called MolGAN, that is a generative framework that will help generate novel molecules in order to predict new entities in the chemical space and help us more explore this space. So the idea here is to see if we can identify new molecular entities that have a drug discovery profile, and we use quantum computing in order to enhance the generative power of existing tool. So our MolGAN QRL model have been achieving up to 16 higher fold in terms of new, valid, and unique molecules that can be detected and provided through the quantum circuits. It have more or less outperformed all existing and compared framework in the literature, and this work is under preparation for publication very soon. So in the nutshell, through the BIND project, we were able to have innovative methodologies through developing new AI-driven pipelines. We have new therapeutic molecules against leishmaniasis, and hopefully we will have similar projects for different infectious diseases in Africa. And we're also doing a lot of capacity building with a lot of new researchers that have been trained in the lab and are forming the new research capacity for our group. So with this, I would like to thank all the team involved, and I would like to thank IC for this opportunity and the African Academies of Science. Thank you for listening.
Thank you so much, Dr. Amna. That was a wonderful presentation of real examples in the lab and how AI is being used, but also you elaborated on another, quantum computing for AI, which was super interesting. So thank you for that. We do have time for 1 or 2 questions, both from online participants or in person. So do raise your hand if you have a question for Dr. Amna before we go on to the next presentation. My colleagues are going to be sending me the questions if we have them from the online participants, or if you raise your hand. So just giving you a minute or 2, well, less than a minute. Yeah. Do you have a question, Vanessa?
Thanks, Emna. I wanted to know what, for the app that you talked about, the CDB, what kind of infrastructure do you need to run something like that in terms of compute or storage?
The models are run offline, so these are real pre-trained models. So it's kind of not that much of capacity that is needed, and it runs instantly. So for predicting, since the molecules is not a big information in there, so it's a small entity, and the model is already pre-trained, so there is no training online that is done. And the idea is really to go onto these type of apps, even for the microscopy, have lightweight models that can be really running very fast and be able to do things online very smoothly.
Thank you. Wonderful. If there are no more questions, I will thank Dr. Amna for her presentation again and go on to our next speaker, who is Dr. Lati Gaye Thiam, Research Associate at Institut Pasteur de Dakar and Senegal. Dr. Lati, the floor is yours.
Okay, thank you very much for your presentation. I'm going to present to you the work we're doing in the malaria vaccine development, and introduce to you about how we leverage genomics and structural biology to help antigen design and get better vaccines against malaria. So, all right. So as you all know, malaria still is a life-threatening disease, and here are some of the latest statistics about the disease, which has claimed lives of nearly 600,000 people in 2023, and most of these were from sub-Saharan Africa, and there is still some more challenges associated to the growing threat of spreading resistance to mosquito and parasites. So this calls normally for more active control measures, but also the development of more interventions such as vaccines, which you all know there are already 2 vaccines that have been licensed or homologated by the WHO, which are the RTS vaccines and the R21 vaccines that you can see here that target the pre-recessive stage of the disease. These vaccines are good at some point because they help reduce the disease burden, but also help prevent severe cases of the disease. But the issue with these vaccines are that they are not protective at 100%. They only protect 30 to 40% of the vaccinees, but this protection is also associated by the strain specificity of the immune response that is triggered by the vaccine. So we need to complement these vaccines by an additional layer of vaccines that can target other stages such as the blood stage vaccines or the mosquito stage. So for the sake of this presentation, I'm going to talk about the blood stage vaccines and how we leverage the techniques that are currently available to help design a better vaccines that will avoid this strain specificity. So for that, we are targeting one complex antigens that is used by the parasites on the surface of the red blood cells. Sometimes people might ask why this specific complex, because the parasites at this stage express almost 5,600 genes, and each of these, most of them can be targeted as a vaccine. But most of these vaccine candidates are polymorphic and can be used as alternatives for one if one is blocked. But one of the specificities of this candidate complex is that all the members are essential. So it means that the parasites cannot do without one of these, and they are highly conserved somehow, and also they induce neutralizing antibodies that can be transcending against most of the clinical isolates circulating on the laboratory lines. So one thing that was not taken into consideration during the design of the first vaccines were the genetic diversity, because most of these vaccines were designed based on laboratory lines that have been maintained in cultures for decades and therefore have lost all contact with immune responses and were not challenged to have some level of genetic diversity. So they were not truly reflected what's happening in the field. So what we are doing in our lab is to bring this vaccine discovery and development closer to the field and start at the early stages to investigate this potential impact of genetic diversity, looking at these clinical isolates that are causing the disease, and that ultimately would be the target of any given vaccine to be developed. So for this, we are leveraging on genomics techniques such as next generation sequencing and also CRISPR-Cas9 gene editing to investigate the involvement of these synapses or polymorphisms in the parasite fitness, but also using structure-guided design to help ameliorate the vaccine candidates on which we are working. And to add to that, we are also functionally testing any given vaccine candidates using real parasites that are collected from the field. So in the next slides, I'm going to show you what we've been able to do over the last few years. So here I'm showing you on the left side of the slide where we did our sampling in Kédougou, which is a very highly endemic region in Senegal, where malaria transmission is like 10 times higher than the national prevalence of the disease. So it's a very endemic region. And there are like very complex parasites circulating from neighboring countries such as Guinea, Burkina, Mali, and others because of the mining activities and the commercial activities of that region. So we sampled these parasites from Kédougou and used deep amplicon sequencing to amplify the targeted genes of one of these vaccine candidates, which is PFRH5, which is the only one of these members that is interacting with the red blood cells. And unlike what have been seen with other sequencing techniques that showed before this study, only 10 synapses that were higher at that were found at percentages or frequencies of higher than 5%. Here we found that in 186 parasites that were sequenced, 90 synapses were identified within this gene, and 70 of these synapses were novel. And within these 70 novel synapses, 47 were present at very low frequencies, as you can see. So this is something that highlights the power of these techniques, which can help you go deep into the sequencing and population and find rare variants, because most of the time these parasites are collected as polyclonal infections, and you may have some very rare parasite genotype that will be circulating but are not picked up by standard sequencing techniques. So that's one of the power of this sequencing technique. So to move further, we went on to now use in silico predicting tools to trade these SNPs onto the structure of the vaccine antigens and predict the functional impact they might have in vaccine development or the antigen structure. So we found that some of the antigens or some of the SNPs could alter the binding of the antigen to the receptor or one of these complex members, which is SIRPA, or it can alter the structure. So by altering the structure, it can also modify the epitopes that are being recognized by antibodies and thereby causing immune evasion, which is something that is not good and that might be causing failure of the vaccine. So the genuine question we asked was, will this variant impact efficacy of vaccine-induced antibodies to the PFRS5 vaccine, which is one of the leading candidates currently in clinical trial? So to answer this question, we went on to use vaccine-induced monoclonal antibodies from 2 clinical trials, phase 1 clinical trials that were conducted by our collaborator in the UK, by Simon Draper's group. And these trials were either a viral vector trial or an RS5 vaccine based on the monoclonal of the recombinant antigen with the AS01 adjuvant. So we went on to collect B cells from these vaccines, sorted them, and used the take the B cell specific, the antigen specific B cells, from which we amplify the variable heavy and light chains of these genes and use them to clone into plasmids and produce the monoclonal antibodies that were further tested for specificity of the antigens. And these were used in silico based ex vivo assays, functional assays. So we went for ex vivo assays because this means that the parasites are put into contact with antibodies less than 6 hours after collection, which means that they are more reflective of what's happening in the body. So this is the reason we chose this approach. And the second techniques we use is the flow cytometry based assay, which will help us quantify high number of cells and discriminate which ones are infected and which ones are not infected, and compare our tested antibodies to the control antibodies. And further on, we analyze our data, and this is a snapshot of the type of data we have here. Here you can see the antibodies that are like labeled based on the specificity and also the strength of inhibition. So we have antibodies that were classified as GIA. GIA means growth inhibition activity. That means the percentage of activity they have in inhibiting the parasites. So we have the low inhibitors, which are less than 40% inhibition, the medium inhibitors, which have between 40 and 60%, and those that are high inhibitors are on the top panel, you can see in red. Those ones can yield up to 80 to 90% inhibition. But we did not see any significant difference between the inhibition of these clinical isolates, which are circulating, with regard to the reference strains, which is the 3D7 strains that was used as a vaccine allele. So this is one encouraging results, because this means that we can overcome what happened with the RTSS and R21 vaccines. So, and this also data match what has previously been shown with other laboratory lines that are from different geographical regions, so which strengthen also this data output. So we want further to test now vaccines against, or antibodies against 3 members of the complex. And as you can see in this figure, there is a kind of gradient in terms of susceptibility of the clinical isolates as we move from the top of the parasites down to the closer to the red blood cell membrane. So you can see that antibodies targeting the RH5 are stronger in inhibiting parasites than those targeting SIRPA and RIPA. But one data I'm not showing here is that though these RIPA and SIRPA vaccines antibodies are not that good when tested alone, when you put them in combination, they can act synergistically and increase the yield. So meaning that sometimes we need to have the best polyclonal responses to get the answer we are looking for. So that's where now this design comes in. So far there are 2 vaccines antigens that have been tested. One is the viral vector and the second one is the protein-based vaccines. And these results show that the protein-based yields higher inhibitory antibodies than the viral vector and necessitate to be proceed with the next clinical trials. And here you can see the structure of the vaccine candidate RH5 with the one that was used in both the viral vector regimen and the protein-based regimen, where you have the full antigen structure with the N-terminal, the C-terminal, and the internal loops that are shown in yellow, purple, and then is it magenta? I don't know. So, and then the blue diamond shape is the core of the antigen. And studies have shown that when you use these ones, the other vaccines are targeting this other part of the protein, and they don't yield or they don't trigger strength transcending antibodies. And we were able to map the epitope regions of these antibodies to know exactly where they are binding and what are the strengths they have. So that's where now the structure guided design comes in. You can see here we can now design the antigen in a way that we can immunofocus the responses we need. So that's where we have the enhance or the RH5 minus the non-inhibitory antibody regions, and which was further stabilized to get result of the RH5/2 antigen, which is currently used as a viral-like particle for immunization in the next trial. And these ones have shown in preclinical trials that they yield higher antibody responses, and these antibodies are more like inhibitory than what was shown presently. And to just sum up with what we have designed so far, these are some of the key messages that we think might be good in translating insight into vaccine, the scientific insight into vaccine discovery and design. So with that, I would like to thank my collaborators and my lab members for this wonderful work they've done, and thank you.
Lati, thank you so much for that presentation. Do we have any questions from the in-person audience or online? And while I wait for that, I might just, you gave us a quite detailed presentation on how the different steps of the technique works. And I was wondering, in your discussions yesterday, I mean, our discussions on these different emerging technologies, how do you see the next step of, also other technologies that can add to what's already happening in your field, in your domain of vaccine discovery?
Yeah, thank you. That's a very good question, which I think I've had some anticipation on it. And I think one of the best technologies I'm foreseeing to be used in my field is AI, because of this like genetic diversity, and we have this like use data, genomic data that has been like collected over years in Senegal that can be used, and elsewhere in Africa, that can be used to model these like selective pressure that are being applied into parasites and help immunofocus or design better vaccines, even if it's not only the RH5 vaccine. It could be like vaccines that target different epitopes from different antigens that are involved in different stages. So I think AI could be a very good like technology to be applied for such design.
Thanks so much. And I guess, regional collaboration, international collaboration, but regional especially, as you were saying, in Senegal and then beyond in the African continent, is super important for the field of vaccine discovery as well. If we don't have any questions, I will go on to the next presentation. I don't see hands up, neither questions in the chat. So you do see the question. Wonderful.
Yeah, there's, I think, Alan Omala who is asking, do you observe the same consistency of vaccine protection against different Plasmodium species if species different occur? Yes, so for PFRH5, unfortunately, it's only like, it doesn't have orthologs in other Plasmodium species. It's only present in Plasmodium falciparum. But for the rest of the complex, like Sirpa and Vivax, we do have orthologs in other vaccines, in other species. And currently what we are doing is that we've been able to design some of the chimeric parasites where you replace the RH5 allele, or the Sirpa or Ripa allele of Plasmodium falciparum with the Ripa allele or Sirpa with from Vivax or Novolecide, and you do the culture adaptation and subject them to these antibodies to see. And the preliminary data that one of the students, Rebecca, has done is showing that there are some level of inhibitions when you use antibodies against Ripa and then Sirpa for those other species. Yeah.
Thank you so much for the question and the answer. Again, thanks again, Dr. Lati, for your wonderful presentation. I'll move on to Dr. Chinwe Chukwudi Uzma, Deputy Director and Head of Training and Research and Training at the Vaccine Research Department of University of Nigeria. Dr. Chinwe is also APTI Fellow at the African Academy of Sciences. Dr. Chinwe, the floor is yours.
Thank you very much for the opportunity. This morning I'm going to be talking to us about the work we have been doing trying to end the neglect of human African trypanosomiasis by shining the light with molecular diagnostics just to improve disease diagnostics and disease detection and surveillance. Because we have a very mixed audience, I would like to start with a brief introduction of human African trypanosomiasis. I know that some people here haven't heard about the disease. It is a zoonotic protozoan disease, bloodborne, that is transmitted to humans and animals by the bite of tsetse fly, although there is recently some indication of sexual and maternal transmission. And while the human disease is known to be going down, but the animal disease is becoming more problematic. And so we wanted to use newer technologies, particularly DNA and molecular technologies, to understand what is happening with this disease, both in humans and in animals. But one thing to note is that there are different species of the parasites which affect the humans and different from the ones that affect animals. But one of the things we noticed when we started this work was that the WHO website of disease data for this disease indicates that they haven't recorded this disease in Nigeria from 2012, except for one case that was detected, that was diagnosed in the United Kingdom. And this was very concerning for us because for an infectious disease, we do not think that this case in 2016 could be a single isolated case. It was just a question of being able to make a diagnosis of the disease. And that case was detected in a Nigerian who was visiting the United Kingdom at the time. And so she had access to sophisticated diagnostic techniques, and they were able to detect the disease. But unfortunately, that didn't elicit any further action from the health authorities. And so we looked into the literature, and we found that there has been various research outputs published indicating that there are silent infections, which are people who are infected but are not showing clinical signs. And there have also been others who are showing clinical signs but were detected in some kind of collaborative work. And none of these from the literature was captured in the WHO database. And so despite all of these publications, the WHO database is saying this disease has not been detected in Nigeria in over 10 years. And so we went to work to look at what is the diagnostic approach for African trypanosomiasis in general. I want you to note that I'm going to talk about the differences between the human and animal disease, but I want you to note that in the animal disease, it's very easy for us as veterinarians to just put a drop of blood from the animal, look at it under the microscope, and you will be able to see whether the animal is infected or not. That's because the animals have a very high level of parasitemia, which is number of parasites in the blood. But for the human disease, the difference is that it is very low parasitemia. And so just looking at the blood under the microscope is not enough to give you a diagnosis. This study actually was looking at diagnostic approaches for animal trypanosomiasis, which I don't consider problematic. But they noted that a single diagnostic method applied at a given time does not unequivocally identify the various parasitology and disease status of a host. And so we decided to apply some of the lessons learned from there into the diagnosis of the human disease. And please note that the human disease is in two forms. One is predominant in West Africa, West and Central Africa, and the other one is predominant in East Africa. The one that is predominant in East Africa is very closely related to the animal disease. And so it has features of the disease that are similar to animal disease in the onset of clinical signs. It's acute, it's hyperacemia. But the problematic one is the West African disease, which contributes to 97% of the total prevalence of the disease. And it is a very chronic disease with very low parastemia, and that's where there is a difficulty in diagnosis. And so we try to look at the ease of having a clinical diagnosis of heart. The clinical signs are in early and late stage forms. The early stage form is very similar to malaria, and there is no way to distinguish it. And because it is a neglected tropical disease, not very well known to the health authorities, they never make a clinical diagnosis. And it's also occurring in places where the malaria is co-endemic. So all of the early clinical signs and symptoms are treated as malaria. And so you have people treating malaria over and over again because they are not actually suffering from malaria. And then the late signs are not pathognomic either. They are combined signs of neurologic signs of different diseases. And so clinical diagnosis is not an option. Now, the case definition for this disease is finding the parasite in the blood or lymph node or CSF, because the parasite gets into the CSF in the late stage, which is why they cause neurologic signs. But it's like looking for a needle in a haystack, because because of the very low parasitemia, you have to look at about 200 microscopic fields to be able to find one parasite. And I doubt which technologist will sit down and do that for how many samples. And so this is not an option. Various concentration techniques have been employed, but still it's very low sensitivity to look for the parasites. And so the diagnosis for this disease has been centered on serologic tests. And we all know the problems with serologic tests. They're not very specific because antibodies are cross-reacting, and especially with places where there is malaria co-endemicity. But there are two particular serologic tests that I want to point out, which are the ones that are used by WHO, the CAT, which is a card agglutination test for trypanosomiasis, and the trypanolysis. These two tests are based on variable antigen types, which are known to be absent in isolates from Cameroon and Nigeria. And this could really lead to the false negatives that they are seeing all the time. And then they say, oh, the disease is no longer occurring in Nigeria. I'm going to talk to you about variable antigen types. So this particular parasite has is notorious for changing its coat, kind of. We call that the variant Sovet glycoprotein. So when they infect and the body is beginning to raise antibodies against a particular coat that they have, at some point they shed off that coat and bring on another coat. And so the antibodies are not able to recognize them anymore. And these serologic tests are based on these antibodies. And so they are really, really very unreliable. Now we decided to look into molecular diagnostics, which is really what is more preferable for most infectious diseases now, because with that you can do PCR, you can amplify some sequences and be able to detect as low as 10 trypanosomes per mil, whereas the limit of detection for parasitologic examination will be about 10,000 trypanosomes per mil, and in most human infections, you won't have up to that. And so we employed these techniques with modern tools that are available at the time. I know that PCR has been around for a very long time, but that's what I talked about emerging technologies. But this technology has not been applied even in these life-saving situations. And so we had to come up with an innovative surveillance and diagnostic pipeline where we combine both serologic tests like ELISA and the CAT, and then we take it up to PCR and sequencing. And with that, we piloted this with in a general hospital, a primary care center, where we just took samples that were submitted for lab examination from patients who came for different things. And most of those patients had recurring fever or malaise, and they have been treating for malaria for a long time. And there has been previous reports of finding the human parasite in animals in that area. And so we took these samples and ran them through ELISA, and we found a lot of positives in one group of samples. And to our surprise, this was a disease that they said they haven't seen in 10 years. And so we also did the card agglutination test. I also want us to note that the variable antigen type that is used for the card agglutination test is absent or low in the Fontaine area of Cameroon and in Ethiopia, Nigeria, which are the hotspots of the infection. And so we went ahead to see a lot of positives on the cards, and with a very high serum titer for the disease. And then we also took that to indirect fluorescent antibody test, where we have the serum coating the parasites, and we had confirmed all of these positives by these means. Then we went on to design new molecular tools, primers for PCR, because some of the old primers that I used, which is why they have not been uptaken very well, was designed when primers were being designed by hand and by eyes and all that. Now we have tools where you can just put in your sequences and then you get the best primers by the click of a button. And so we had to redesign new primers to identify both the order and the genus. And then we also designed more primers for the trans ITS, with the full-length ITNS, and we applied these things to the diagnosis, and we were able to confirm that those things that we saw were true positives. And so I want to take a bit of time to talk about the uptake of these new diagnostics. Up until now, the WHO relies on trypanolysis, which is one of those antibody tests, and it is only done at ITM Belgium. So for every place, this disease happens only in West Africa, but for every case that is detected, you have to send samples to ITM Belgium, and the process of sending that is what is limiting the disease reporting. And it brings the question, can African scientists really make adequate diagnosis for African diseases? I think the capacity is being built, and by the help of AAS and the APTI program, we have set up a very high molecular center to be able to do these things. And we wonder why it is difficult for them to take up these new technologies to embrace PCR diagnosis for this particular disease instead of depending on this. But we understand that there's a slow process of reviewing guidelines and policy changes, and there might be some superior diagnostic interest. I'm sorry to say, but that's the truth. So policymakers are not in sync with research and development of new diagnostic techniques. And I want to particularly point out the case of an ongoing case of a 17-year-old with clinical signs of stage 2 and with positive results on all diagnostic tests. And then WHO has gone ahead to do trypanolysis. And based on that trypanolysis, we had this response. The samples are negative with trypanolysis, and therefore no further testing for heart is necessary, and of course no trypanocidal treatment is granted. But these samples were tested with ELISA, CAT, PCR sequencing, and it's all positive. I don't know what we will take as an indication of the thief breaking into your house, a broken window, or a fingerprint of a thief. We did the fingerprinting with DNA, and we saw the parasites, but they don't want that because they are looking for a broken window. I don't know. So in summary, I want to say that molecular diagnostics can improve disease detection. And in this case, we have seen that prevalence have been recorded from 0 to 10% in the population. And we're even beginning to see the disease beyond the known heart foci, where they have been traditionally looking for the disease. And this data suggests a high burden of undiagnosed and unreported clinical heart cases in Nigeria. And we really think that the attention should be there. But unfortunately, the health authorities are not taking up these technologies. It is very abysmal. Maybe we need to engage more in policy advocacy. But the bureaucratic bottlenecks are hindering this disease production. And this continued propagation of the culture of neglect for this disease has very grievous consequences for disease reemergence and epidemics resurgence, which we can talk about. And with that, I want to say thank you for listening, and to all the people that supported this work. Thank you.
Dr. Chinwe, thank you for the super interesting and wonderful presentation. I'm going to turn to the room here to see if anyone has questions. I do have a question. You mentioned that the center that you're doing, so the molecular diagnostics, And if I understood well, so it's really looking at those technologies, as you said, PCR is not used enough, basically. We have it. It's quite accessible in the sense as a technology. It's not too expensive, and it doesn't require too many things to a certain extent. I was wondering if you can elaborate a bit more about how you're doing that in terms of highlighting how you can use certain technologies that exist in probably most basic laboratories, molecular laboratories, and how that they can be used for something so fundamental when it comes to misdiagnosing really important missing the diagnostics, which is an important problem.
So because this disease is classified among the neglected tropical diseases, I actually say it is neglected among the neglected. And so people don't work on it because there is no motivation to do that. Apart from me and the staff of the Nigerian Institute of Trypanosomiasis Research, which has the government mandate to research on that disease, I don't know any other researcher in Nigeria that is working on human African trypanosomiasis. There are lots of people working on animal African trypanosomiasis. The veterinarians are doing amazing work with the animal disease, but nobody is working on the human disease apart from NITA. And the people of NITA are doing more of research consumption. They have an output, they are giving guidelines, they follow it. That's all they do. They don't really do much of basic research into this disease. And so when I started working on this during my postdoctoral training at the NIH in the US, which was sponsored by the ESS APTI program. And with what I saw, I felt like this is not something I could keep on studying away from where it is happening. And so I relocated to Nigeria with the aid of EAS and the grant, the APTI grant, and I also had a container load of equipment donated to me from the NIH just to continue this work. We set up this fantastic center called the Vaccine Research Center with various molecular labs to look into this, and we are also building capacity, training students. Currently, we have about 10 postgraduate students that are being trained on these technologies so that we can amplify the use of this technology. Molecular diagnostics is very expensive, and it requires a lot of expertise, which is why many people don't trust it because they are not sure of the people who are doing that. But I got the training from the best of the places. I have a PhD in molecular biology from the Royal Vet College, University of London, and then a training at the NIH, in the National Institute of Allergy and Infectious Diseases. And so with that, I am very well equipped to train the next generation to apply these technologies to basic disease detection in Nigeria and in Africa. Thank you.
So much, Dr. Chenwe. We do have a question. Yes. Can I just raise this question and give you the microphone? Okay. So we have, can researching on neglected tropical diseases give new insights about new or emerging vectors and their transmission routes?
Absolutely. I think because when I started working on this, I realized that the very basic research that we take for granted in so many other fields, like in malaria, have not been done. The very basics have not been done. And so one of the things that was brought up by the case of this 17-year-old boy, who we have investigated for about 3 years now, was the impact of the transmission routes. Because right now, this boy and all his family were positive in all the tests that were conducted. And we have dug into their medical history. We saw that they all had the mild chronic fever at birth, all the children. And then one died with a very marked leukocytosis, which is a hallmark of this disease. But nobody paid attention. Even for this boy, he has cysts in his brain. And all the medical officers want to do is to open his brain multiple times to take out the cysts one by one, because they don't know what is wrong with him. And with this technology, we've been able to see what is wrong with him. They all had that disease. And we think that it's a case of sexual and maternal transmission that is going on, because the mother was passing it on to all her children, and one died because he couldn't survive the disease. And so you see, many of these people in Nigeria and in many African countries, like the end-stage disease, the late stage of the disease, has neurologic signs where somebody is incoordinated in walking and talking and things like that. And people would generally classify them as mad. It's either it's madness or witchcraft in Africa. That's what we will say. But these people are sick with a very basic disease that can be cured with one card of tablets. But nobody knows what is wrong with them. And they're being treated for everything. This boy has been placed on anti-epileptic drugs for over 4 years with no improvement. And now it is neurosurgery. And so we see that there are lots of lessons to learn, but because people are not even uptaking the basic technology that can help us to learn those lessons. So with molecular diagnostics and with molecular studies, we can actually learn a lot more about African trypanosomiasis.
Lot, thank you. The question here in the room.
Okay. Thank you very much, Dr. Chinwe, for your excellent presentation. Mine is more of a comment. Just like the previous presenter, listening to your presentation, I see more opportunities for applying emerging technologies to be able to grab and prove some of these points beyond reasonable doubt, like the application of AI in the disease detection and as well as in treatment. But most importantly, as a policy advocate, this is another critical case of global politics in science, where sometimes the research outputs from the global south are not regarded as genuine and are not also publishable, because I believe that if you send your article, an article coming out from this research to a world-class journal, it may be rejected instanta because they don't think it has followed their own pathways to impact. So this calls for greater awareness and advocacy, especially coming from the Africa side. I'm happy that we have African advocates of science in this room. My organization is one, the ATPS, the AAS, and when we meet at the African Union STI committee, this is the kind of cases or a case that we need to raise, that the global politics in science should also not affect, in agriculture we call it like orphan crops that they do not take interest in because it may not be yielding enough cash for the economies, but it is something that sustains a people, a community. So there is greater need for advocacy to listen to some of these research outputs coming from using molecular diagnostics that have been applied. But like I said, in summary, there is need to apply better technologies, AI technologies that can nail this thing without much ado. Thank you very much.
Yeah, thank you very much for this very important comment, and I think we all take it to heart as our
Sorry,
role.
1 second. Let
Yes.
me quickly say that just while I was here, my a paper that we sent to a journal was rejected for the second time without review. So he's very correct about that. You cannot publish any of these things because the system is set against you. And as soon as you're sending the publication, they are rejecting it without even giving an option of review.
Maybe.
Just
Ca-
as an
ca-
addendum
yeah.
to that, sorry, moderator. You see, I've had similar experience where I come up with some findings and I send to some journals and they reject it. So there is always an opportunity. There are a lot of African science, technology and innovation journals. Look for such and get it out first. Let it be counted first. Whether it has 10 impact factor point or it doesn't have impact factor point is not the case here. but let it become one of the knowledge products that has been published. And people can begin to cite it. You'll be surprised that the citation will be so high that they will have no option than to take up similar, accept similar results in your next findings and publication. Thank you.
I think we can discuss a lot longer on these issues for sure. Can I keep it for later after if we still have time, or when we go to the coffee break to continue the desk questions? Thank you. Just in sake of time to be online as well with the participants. So our final speaker is Professor David Dodo Aran, Director of Research and Innovation at the University of Ghana, and also Fellow of the African Academy of Sciences. Professor David, please go ahead.
Okay. Thank you very much to you and the organizers. We've listened to the health aspect, and what I'm going to discuss with you as I wait for the slide to be up is from the material science and engineering perspective, and how we can relate these ones to waste management and putting emerging technologies into waste management that is directly associated with local communities. So the title of the talk here is turning our waste into fuel. So what we are looking at is converting plastic waste into fuels that can be used by local people. But basically, this is just a brief one from where I'm coming from, Ghana. If you've never visited, please do. From the top there, I'm sitting on the crocodile. You can come and have an experience of this friendly country. Also, I'm from the University of Ghana. Sorry about that. Okay, so this is University of Ghana, where I'm coming from. I'd also like to take this opportunity to invite you to a meeting that we are having in December, if you happen to be in Ghana and then pass through there. We have various sessions that we'll go through, and I invite you to do that. Going forward, we're looking at waste management, and waste management, as we know, is a global challenge, especially in the global south and most part of the African continent. One of this is the plastic waste, which has become a lot of menace across the continent and in some part of the world. But we know that there are a lot of emissions that are resulting out of these wastes, so we can have a reduction of about 15 to 20% of the world's greenhouse emissions that could be taken off through sustainable waste management. We also know that as of 2020, over 100 million tons of these wastes were generated worldwide. And then we found out that about 34% of the total plastic markets were generated, basically being from plastic and polyethylene resins, and the numbers keep going up. This calls for a reason for us to deal with this problem. Okay, so there are global trends. In Africa, the number is still going up. So many countries are already unable to deal with the amount of waste in terms of the plastic waste that is generated, and only a fraction is usually recycled. And then we have most going into the landfill size, so only about 9% of the 9 billion thereabout tons of the plastic waste that are generated are recycled. So here means that there are challenges. We still have landfill-sized dumps and these plastics going into the ocean and also into the environment. So this calls for reasons for considering things like waste recycling and then adding value to these ones. So from a various perspective, we can put them into construction. We can also put them into the road construction. We can also look at converting these plastics into fuel, into what we call energy. So in the recycling, you can think about reduce, reuse, or we can also recycle. But our focus here is to look at how we can valorize these waste. We can make money out of this waste. So that's why we are calling it waste to wealth. So even though it is waste, we can have some economic benefits to this, which will be an incentivization for the reduction of these. What did we do? In the waste conversion to energy technologies, there are various actors within the value chain. So we have different plants that have been set up to convert these waste. One of them and the technologies that we are looking at is called pyrolysis, where we are able to convert these plastics into various forms using heat, using energy. So what we do is the pyrolysis process of the plastics, when you heat them up, you're able to break these plastics into gaseous forms. You can also have liquid forms. You can also have solid residues that come. This liquid residue is in the form of diesel. You can have it in the form of petrol, and we can put these ones into various applications. What we have done is to convert these plastics and then put them into real use in vehicles, and then also into outboard motor. The reason for doing this is that we want to help solve the problem of marine debris. What is going from the land, getting into the oceans, this is becoming a canker. So we should find a solution. So we work with communities along the coast where the plastic debris actually enter into the ocean. By incentivizing them to get fuel for their outboard motors, the fisher folks who do collect the plastics as plastic fish can convert this into fuel that they can use for their outboard motor, and then it could be for sales. So these are some of the products that we can get from this. You can get you can get char, as we call charcoal, that you can use for various applications. We can get gas, and then here are some of the liquid products that we have already received. So we have done a basic kind of research at the University of Ghana. We did a demonstration. As we can see here, we built a locally made processor, which we call the metal reactor that we use for the conversion. So here we did a project that was funded by the French embassy, and we produced the fuels. And here, as you can see, the real use of the fuels into vehicles. So the last one, as you can see, this car runs on plastic fuel. And then we did a demonstration, and then it actually works. Based on this, we had to do an extension into communities. So we entered into vulnerable communities where we have the plastic waste entering into the ocean. So there's a place called Osu in Accra, close to a heritage site. So it's one of the UNESCO heritage sites area. And you have all these plastic waste entering into the ocean. So we have done this, whatever we did in the university, we brought it to the community. So we had a lot of community engagement. And this project actually trains the local youth in the conversion of the plastics, and then bringing them into the collection, the value chain, and then using all these ones. So we have a whole setup that we have done, and then we show here some of the fuels that have been produced. So we also did a production of these fuels, and here is the demonstration of locally made systems for converting this plastic waste into the various forms of the fuel. So we did this, and then there's the training. Now we go directly at the beach. So as you can see, the one doing the demonstration, when we look at the picture that is on the first side, and we gave this fuel to the fisher folks, and they put into their canoe. So for us to test and be sure that the fuel that we have produced is indeed good, I had to be on the boat. So else they don't believe that it is real product. So what we did was to use their own canoes, to use their own outboard motor, and then we took the fuel. So the other side on the top is where the fuels that we have produced, they pour into the gallons, and then we use. So as you see from the below on my left, when we were sitting on the canoe, then the last part is when I was actually on the sea. So we did about 30 minutes deep into the sea, into the horizon, to see how the performance of the fuel was. At some point, they left it if it fails. But fortunately, the fuel tend to be of high quality than the one that they already use on the market. So now there is a high demand for us to go and then replicate some of these along the various coasts. So how do we bring our emerging technologies and possibly the AI into these systems? My concentration has been with these vulnerable communities and then trying to bring some solutions. So it's a dual solution. We're removing the plastics. We still have a lot of the plastic waste being generated, and we should still find some solutions. And then train these local communities to be able to do the conversions themselves. But after doing this, we have to think about scaling up. So that's the next phase of going on to scaling up. In scaling up means that we have to think about some technologies that are for helping with scalability. We have to think about more efficiency in terms of these ones. But the concentration here is integration of artificial intelligence and machine learning into our processes. So what can we do? So we're thinking that we can automate the system. We'll build a system along the coast where these debris are entering directly into the ocean. It means that we have to do identification, separation of the different forms of the plastics. We have all these mixtures that we have, meaning that we can also install some sensors to make the process of the sorting also very good. Then we're thinking of artificial intelligence predicted material flow and AI incorporated in the system design for all these ones. In effect, we want to have energy efficient and then cost effective approaches. So in conclusion, yes, I would like us to also still take this opportunity to invite you to the meeting that we are having in Ghana. And we believe that with these solutions, we can better help our local communities. Thank you very much for your attention.
Thank you so much, Professor David, for this wonderful, demonstration of an example that is so on ground and directly working with the people who are going to help you use this technology and this transformation for something so important. We do have a question from online, and I'll ask the in-person participants to think about their question if they wanted to. We have 5 minutes. So the question is, how did your team ensure health safety in these plastic processes communities as we anticipate toxic byproducts that may be exposed to workers?
Okay, so in this whole process, which I didn't go through in detail, we have what they call a circular economy. So this whole design is such that we do not allow emissions to come out. So the processor is built such that the gases that are generated are collected through a cleaning system. It's just like the car exhaust that you have a catalytic cracker in. So we go through some filters and we filter the gas that is produced. Then these gases that are produced are collected, compressed, and reused. So you have a circular system that is coming in. So in this project, from material science and engineering perspective, we had to design ceramic filters. So we use local materials to design ceramic filters that we put in that does the cleaning. So you can remove all the NOx and all those ones. And then the system is such that you don't have emissions coming out. Obviously, with the participants, they have to use the mask and then all those things in case there are some fuel leakages that might happen. But before we were allowed to do this, we had to go through the ethical clearance, you need to environmental protection, coming in to make sure that the emissions levels are really controlled. So part of it is to have emission sensors in the work area to see if there are other emissions that are coming out as a result. And we collect this gas, and the gas is just like liquefied petroleum gas, so it can still be used. We did a testing using the gas. Then the various liquids, depending on the conditions that we have, you can have petrol. So we know that if you want diesel, we can have diesel under a certain condition. If you want petrol, we can have them under guided conditions. And that is basically how the process has been.
Design and checks throughout. We do have a question from online, from in person. I'm going to guide the camera to our participant who will ask her question.
Good morning once again. I'm Bridget Mutuma and a Rice Fellow. Thank you very much, Professor David, for your presentation. I have a question and a comment. So the question is in regards to the standardization of the fuel that you generate from the waste. So do you generate the fuel from one type of waste or from a mixture of the plastic waste that you've collected? And then for the comment, my work is on plastic waste, but I look more into the microplastics and the nanoplastics and their detection. You're making sensors and other adsorbents to try and capture them from water and in the environment. And I'm thinking in line of how you can also look at that approach and possible collaboration to diversify not only fuel production, but also in terms of environmental monitoring and remediation, looking at the smaller aspects of the plastics, and also trying to leverage AI in terms of that. Thank you.
Thank you very much. So in this work, what we did was to do the various sorting of the plastic, so we don't put every plastic together. It's key to mention that we didn't do that of PVCs because they already have their own challenges. The focus of this has been on single-use plastics, basically the PETs, the PEs, the single-use, all those ones. We do sorting, so we don't put everything together. It could be put together. Then you can get what we call the refuse-derived fuels, depending on the level of the quality that you want to have. That is why it is important to help with the sorting process with AI to be able to sort. Sometimes on the market, it's just to look at the type of the plastic to tell whether this is PET, but sometimes you cannot easily tell whether this is actually PET or another form of it. Some use colorization, and then they can have all these effects. That's where the AI comes in with the sorting and then separation, so that it can fix these parts. We've done also for vehicular tires, car tires, and then we can use the same process to generate that. With the microplastics, it's one of the reasons we are doing this. So we're looking at land to sea plastics. So to have microplastics, it means that you are going to degrade, you're going to break these structures of the plastics into smaller units. And typically when they enter into the ocean, based on the various activities, you tend to have these plastics becoming micro level that are ingested by organisms. We've done some work with the Marine and Fisheries group in Ghana. We have a publication that is already out there. There we found traces of microplastics in oysters. And it's kind of work that is going on. It means that this is still a very serious problem because the plastics are now entering into the food chain, and we need to deal with it. So for me, it's an open, collaborative call for us to be able to work together. Thank you.
Much, Professor David. And for the sake of time, I will have to end here. We do have 4 questions online that I'm going to relate to you so that we can tell the participants. There is one thing I would like us to highlight here, because someone is asking about the event in Ghana. Can you just say quickly when that's happening that you mentioned, and then I'm going to wrap up the session.
Okay. The conference is happening from the 9th to the 12th of December 2025 in Accra. And is the Materials Research Society Conference, and it cuts across various discussions that we have done here.
Okay, very good. Well noted. So with this, I'm going to wrap up and thank all the 4 speakers for your wonderful presentations. All diverse, we got great examples and really good questions. So thank you for the online participants and everyone here with us in the room. And with this, I close this online session. Thank you all so much for joining us. Goal was to announce that we have a coffee break now. I was wondering why no one's moving.